Hi, this is Stanley Maloy. I'm here with MicrobeWorld at the American Association for the Advancement of Science annual meeting here in Washington, D.C. AAAS is the oldest and largest general scientific society in the world. This meeting covers all areas of science, from astronomy to chemistry, physics, psychology, you name it, it's here. And of course, there's some very excellent microbiology being presented at this meeting. So we're going to interview some of the scientists who are speaking at this meeting. Let's begin with an interview with Professor Beatrice Hahn. Beatrice is a professor of medicine and microbiology at the University of Alabama Medical School, and she's done some really groundbreaking work on our understanding of how disease moves from animals to humans. And you were involved in the very early studies of tracking how HIV came into the human population. Could you tell us something about that?
That's correct. I worked with Dr. Gallo here in Washington, actually, as a postdoctoral fellow on HIV when it was first discovered in his laboratory, and I was always interested where this epidemic came from, out of the blue, it seemed at the time. And of course, in collaboration with many other colleagues, we realized pretty soon that HIV was a primate lentivirus. And in fact, there was an early clue because there was a second AIDS virus that was discovered very soon thereafter. And when we compared the genetic makeup of these two viruses, they were not similar, yet they caused the same disease. And they fit into a group of viruses that we call primate lentiviruses. And so from the very beginning, it was clear that HIV must have jumped from a primate source to humans.
Now, recently, you've also been doing some studies on malaria and how malaria originated and came into the human population.
Yes, so one step back, the way we actually traced HIV-1 back to its ape origins was by doing non-invasive studies. We realized the closest relative to HIV was a virus infecting chimpanzees. And obviously you can't study chimpanzees in the wild invasively. It's unethical and also impractical. So we developed methods that allowed us to detect the virus in wild living chimpanzees and then later gorillas. As it turns out, gorillas were also a source of HIV-1. There were actually 4 transmissions, only one of which resulted in the global AIDS epidemic. But in order to do that, we looked at fecal samples. And fecal samples actually give you a lot of information. We could look for antibodies that would indicate infection. We could also look for nucleic acids that we could actually take the virus out of the fecal sample and look at it genetically. And that worked well for tracing the origin of HIV-1. And more recently, other investigators found that you could also amplify Plasmodium, which is, of course, the cause of malaria, Plasmodium parasite DNA out of fecal samples from apes. And so we had this collection of fecal samples just waiting to be tapped for that, and so that's how we got into the origin of malaria.
So where did malaria come from?
Malaria came from Western gorillas.
Now, in both of these cases, in the case of the HIV, SIV virus, and in the case of malaria, is there a lot of disease burden in the animals in nature?
That's a very interesting question. In the case of SIV, the simian version, chimpanzees actually develop AIDS just like humans do. And that was long unknown because wild chimpanzees cannot be observed except for one place, and that is in Gombe National Park. Jane Goodall's famous chimpanzees, they happen to have SIV-ZPZ infection. And so we've been working with the Goodall investigators for over 10 years, having prospectively followed these chimpanzees, and some of them are infected, and we had an opportunity to look at their life history. We had some who died, either infected or uninfected, and that allowed us to actually determine that the virus does very similar things in these chimpanzees. Of course, it takes 10 years and longer, just like it does in humans. You can't discover this on a minute's notice, but these long-term studies allowed us to show that AIDS is not a new thing. It was in chimps long before it went into humans. With respect to malaria, we simply are too early. We suspect that just because the Plasmodium parasite burden in wild apes is so enormous, they wouldn't, if there was a big disease associated with that, they couldn't survive. So we suspect, again, that wild apes are like humans in hyperendemic areas where adults are doing fine, but kids are the ones that are dying, and we suspect that may also be the case in the wild ape populations, but that's speculation right now.
So this is really interesting because it gives us a picture of how the disease evolved, how it came to be in humans. Can you, in any of these cases, track down what the change was that allowed the major jump between species?
Yes, for HIV and SIV, we actually have a pretty good clue. And this is not my work. This is actually work by a number of very talented groups here as well as in Europe. Turns out that humans and other species have innate proteins that are designed to protect them from such cross-species jumps. And there are a variety of those in humans. They have exotic names like ApoBec and TRIM5-alpha and tetherin. And people have shown that these are designed as part of the innate immune system to protect against invading microbes. It's an unspecific effect. So for example, tetherin just tethers enveloped viruses on the surface of cells, so they can't get off, and they get dragged back inside the cell and eliminated. And so turns out that tetherin probably had a big play in which SIVs could jump into humans and which ones could not. Because as it now turns out, as we follow, since we now pretty well know where not just HIV-1 came from, but even the chimp and gorilla precursor of HIV-1, they originated in monkeys, we can trace which genes were responsible for counteracting tetherin and how that changed as the virus moved from monkeys to apes and from apes to humans. And as it turns out, the human tetherin has a deletion in a critical domain where usually viral counteractions occur. And so when SIV from chimps and gorillas jumped into humans, it couldn't, the protein that usually counteracts the human restriction factor couldn't work anymore. So actually another gene had to be recruited to take over that function. And of the 4 different jumps to humans, only 1 made it. And that's the one that caused the AIDS pandemic. So we have a pretty good idea what is sort of restricting these jumps. And the more we can learn about that, the more we can prevent future such outbreaks.
Now, one of the things that you have really used in your work is deep sequencing, and more recently, single genome sequencing. It's getting a lot easier, faster, and cheaper to use those types of technologies. How do you think that's going to change the field?
Oh, it's going to revolutionize the field. And it's really an exciting new area of molecular studies that opens doors you couldn't have imagined 5 years ago. So for example, deep sequencing, we are using it right now to ask the question, just like SIV jumped to humans and caused HIV, and now we know that malaria comes from Western gorillas, are there ongoing transmissions from wild apes to humans in areas where humans and apes live in close proximity? And from our work in HIV, we know the jump alone isn't sufficient. You need time to adapt to the new host. There is a hurdle that you have to overcome. And so it is possible that, for example, with malaria, that jumps occur, but they are sort of camouflaged by the regular human malaria that's already there. So the way to address that is to go to a person who has malaria and do deep sequencing to go beyond the regular Plasmodium falciparum infection that these people have, to see, is there something under the surface in a lower frequency that could percolate along and eventually move up. And that's how we're using deep sequencing right now. I don't have the results yet, but that is what we're doing.
Well, that's a great story, and there's a wonderful future to study a lot of other diseases, and as you said earlier, maybe be able to prevent new pandemics before they happen. So
We would hope so, yes.
thank you very much for
Thank you.
talking with us
Thank
today.
you.